I am, as Alex said, I am the core manager at Moffitt Cancer Center for the flow cytometry core, and I'm very grateful for this opportunity to share my Tyto experiences with you. Just as a overview to make sure everybody knows what we are, who we are, what we do. We have been a shared resource facility since 1990 at Moffitt, one of the first ones that was created. We are a centralized facility with cutting edge flow cytometry services to support the cancer related research of our members and their trainees. We provide highly skilled personnel and state of the art technical assistance with flow cytometry analysis and cell sorting. And like most core facilities, our specific aims include delivering that expert assistance in experiment design and analysis, providing those state of the art technologies and services. And one of my passions is providing the training and education, which you'll see later is a major component of this new cell sorting technology. As far as our staff, we're very well rounded. We have a lot of experience. We have a lot of different roles that are played, both scientific and administrative. And I feel like we have probably the best core staff of my experiences here in nearly 29 years at Moffitt. Not to go into a lot of details as far as services, I'm sure if you're a user, a core manager, or a core staff person, you've probably been exposed to one or more of these types of services in a flow core. What we're going to focus on today is, of course, our cell sorting and what we provide as platforms. So, not to get into too many details about the other platforms, that's not what today's about, but giving you a kind of a starting point because I was asked to provide our journey to this new technology. I want to go back to the start. So, again, if anybody's had any time spent in in a flow cytometry facility of any type, there's probably some sort of legacy instrument, including an electrostatic droplet sorter, which you'll see on my slide here. We have one of the most common ones, our Fax Aria, uh, housed in a big BioProtect custom BSE for the purposes of safe sorting and protecting the users, operators, and of course the sample indirectly. But these are the traditional types of sorters that I was exposed to way back in the old days. And for the most part, were the majority of the services provided in any kind of laboratory that had cell sorting. We also in our lab have a Automax Pro magnetic bead sorter, again from Miltenyi. Um, we use this very sparingly these days because of the advances in our technologies for cell sorting, but it still has utility. It can be used independently as a single epitope isolation device. It can be synergistically used with our droplet sorters. And now we're finding it can be used along with our microchip sorting capabilities. It all depends on the outcomes, the desired outcomes in the downstream assays that our users of a wide variety are intending to achieve. So we all that those of us the royal those of us who are experienced in droplet sorting, we understand the pros, and specifically with our aria. I've enumerated them here. I'm not going to read the slide to you. You guys can read it for yourself. But effectively, what in a nutshell, this instrument has been a workhorse over the years. It's been on our bench since our first one was installed in 2004. So we've been using this technology very routinely. It's a fan favorite. It is always going to be at least as far as I'm concerned, until I retire from this business, it'll probably always have a strong platform usage in our facility because of the pros that are afforded. You can do complex panels because of the optical bench flexibilities. It's a good high resolution signal processing capability. It has automated features for QC and sort setup. It can sort multiple populations, etc. And it can support a variety of different types of cells and sizes with the different types of nozzles. But for every enumerated pro, if we're all being honest with ourselves and with our users, these droplet sorters also have a series of pitfalls. We've until probably early 2020, we weren't really seeing publications on this. So we were communicating amongst ourselves. We were chitter chattering about it, but we knew a lot of these pitfalls were already part of our process because we were getting the feedback from our customers. Hey, I sorted these X type cells, whether they were primary or cultured or whatever they were. We would always have that one customer that come back and say, "Hey, my viability wasn't what I expected after the sort," or they didn't recover fast enough, or my genomic studies, the downstream studies were kind of anomalous. They weren't what we expected, or we had some variability in our longitudinal studies. Whatever the reasons, we knew these pitfalls from these instruments were contributing to those problems. And again, I won't read the slide to you, but you can see where these can be problematic. One of the biggest problems, and you'll see this later on, is the external fluidics that are required in most of these droplet sorters are not necessarily compatible with the biology and the metabolism of the cells that and when you're in a core facility, you work with a wide variety of them. Not everybody's sorting T cells, which are, in my opinion, the hardest ones to sort. In a cancer center, you're sorting some pretty fragile cells in some cases, and those buffers that drive the instrument performance don't necessarily gel well with these cells in some cases. Another thing that's really, really a standout for us is the complexity of these instruments and the amount of training needed, whether it's a new staff person in the core, or it's a new user, or a user that is doing a lot of sorting and really wants more independence and more access. It can take a while to get that person safely trained, competent, and really ready to be independent. Okay. So again, I was asked to provide some background. How did we get to where we are now with the instrumentation on our bench and the access that our customers have? So some of the details of the beginning exposure to the are fuzzy in my head because it's been so long, but I'm pretty sure I can confidently say the first time Miltenyi revealed it was in Cyto 2016 out in Seattle. It was the big reveal, and if you can tell from the picture, it was a big reveal. Kudos to the marketing team. The booth was fantastic. It was visually flashy, fun, eye-catching, and certainly attractive. From my memory, most everybody spent all their time at this booth. And who wouldn't want to come, party with these people, especially this beautiful mascot, a Tyto mascot that paraded all around the meeting that year. So it was a pretty interesting reveal, and it certainly got me thinking about it as a core manager. But apart from all the glitz and glam, I stepped back away from the moment, and I had to be honest to say, what do we need? What do my customers need? What do we do now? And what are we going to be doing in the future? And how would this technology support that? And I have to be, thoroughly, honest and open. For the first 2, 3, maybe 4 years after that big reveal, I was like, "No, thank you, not yet." Because I wasn't confident at that time that Miltenyi was gonna be a big flow cytometry player at just yet. They were there, they were getting there, but for me, I wasn't ready to take that leap. Microfluidic based technologies were starting to emerge, but they were so new and untested. And I'd been through plenty of piloting, testing instruments in my experience. I didn't think it was ready for prime time just yet. Let's see how it goes, right? Let's not jump too quick. But most importantly for us, I knew we were transitioning away from a lot of the immunophenotypic cell sorting projects into these big cancer cell lines. We were doing a lot of transgenic uh, models. We were expanding cell lines. We were getting into the CAR T space. Those immortalized cell lines are much bigger And the one sticking point for me was the cartridge with that 20 micron embedded filter. I'm thinking, nope, those big basketball sized cancer cells are never going to go through there. So I took a little bit of a, you know, pause and said, let's wait and see. But at the same time, what started happening, and I'm not going to read these slides to you either. You can go back and look at these references, but they came out around, you can see the date on the one on the left here, April 2021. Um, uh, uh, About here, this was 2020. People were actually starting to evaluate and publish what we have come to learn is sorting induced cellular stress, or appropriately acronymed SIX, which was the direct impact of these droplet sorter pitfalls on our cells, on not every type of cell, but there were a variety of different cells tested and published on that we absolutely could see there were some distinct differences and problems that we had to be honest with our customers about. And even the ones that were coming to us and saying, hey, we have problems, we would talk to them about these potential problems, but now they were being enumerated in publications. Keywords like metabolic perturbations observed in NIH 323. How many times did I sort NIH 323 cells on my ARIA? Um, Wide range of functional defects. Again, sorter induced cell stress, differences between individual experiments on the same instrument. So, trying to do longitudinal studies, if you're getting differences in your results, you can't make conclusions. So I had to go back and start thinking, all right, let's reevaluate the important features that my very persistent and persuasive salesperson and colleague was trying to impress upon me. I'll never forget one of the first things she said to me when she took this role. She said, "I will put a Tyto in your lab." Now it took her 18 months, but after really looking honestly at these features that I felt would directly improve our cell sorting services, I said, okay, it's time to put these in a justification and a PowerPoint presentation for my advisory committee, because this is a leap. This was back in 2021 to 20 or late 2021, early 2022. I put together this presentation and said, listen, guys, I know this is new technology. It's difficult to get early adopters in the South. I promise you, I've run this core long enough to know that it's a field of dreams kind of mentality. You build it and they'll eventually come, but it's going to take some pretty good coaxing. So I presented these arguments and said, okay, granted, it's definitely new technology, but if you give me a shot, let me see if this technology can help me help you. And they gave me two thumbs up each. And within 18 months, we said yes to the MACSQuant Tyto, and we installed our original version of it in March of 2023. And about the same time, right next door, in the Journal of Proteome Research, this group published their direct comparison results. They were comparing their several cell lines sorted on their ARIA and sorted simultaneously side by side on their Tyto. And again, you can go look this. It's from 2024. You can go look this citation up and get all the juicy details. But things that in the abstract that stood out to me, microfluidic chip-based sorting has a minimum impact on metabolism and is less disruptive compared to the droplet-based sorting. And I pulled out of that the major bullet points of high pressure charged droplet sorting versus the microfluidic sorting. And in a nutshell, the droplet sorter had significant metabolic and phosphoproteomic changes, and the microchip based samples, they more closely resembled the unsorted control group, recovered their mitochondrial activity quicker, and there was far less impact on the amino acid and lipobiosynthesis. I mean, I don't know how much more evidence you need to convince me, meaning me. How much more evidence do you need? right? If you know these things are out there and they're being published and proven, you got to be a responsible core manager and you got to at least give this a chance. So we did. And so immediately we we adopted it and we took our role very seriously and we with us within the staff tapping my very intelligent, bright, and young, energetic, self sorting go to staff member, Bethany here. She and I developed a workflow so that we could support in an assisted capacity, similar to what we had done with our droplet sorting services. We developed a workflow to introduce this to our customers. And again, I don't want to read the details of the slide. I want to make sure that Gert has plenty of time to share his experiences. But in a nutshell, we found this four-step process to be very quick and easy, much quicker and easier than our droplet sorter process that we've been doing for several decades. And we looked at each other after about a month of this and we said, this has to get disseminated in a customer training so that we can provide a broader access to our for independent users to be able to have the same experience. And so very quickly, the two of us developed a 2 session training that took that workflow on that. For the first session, we do a instructor demo of the same exact steps that are enumerated here. um, it took literally about a half a morning to do a training demo, and then either the same afternoon with that person's actual sort sample or the next morning or very soon, encouraged to be very soon afterwards. We took the second session and let the user do all the hands on with a little bit of supervision from us. Once we were convinced as a team that the person was competent and capable and confident. They were certified for independent usage. And because of the user friendly interface and the simplicity of the workflow, they're certified 24/7. They literally can come in my lab, and there are people who do. I call them my ghost sorters. They'll come in before our staff even arrives for the morning, and they'll come in long after we're gone. They'll get their sorts done, and we don't even know they're there until I bill them. So if and I promise you, because I got a slide to prove it, we have definitely incorporated this technology as a useful tool that is a fan favorite, and now we have proof and convinced our advisory committee to get a second one. Okay, if this usage slide doesn't tell you all you need to know, I don't know what other compelling information I can share. The first year we had it wasn't even a complete year, but it still ended up with 24% of our cell sorting services as compared to the two ARIAs that we had in the lab. But, it takes some time to get those, suspicious, skeptical minds to be convinced. And word of mouth is very powerful. What happened during that less than a year time? People who were ready to take the plunge were having very successful, life changing experiences, and getting the word out in testimonials internally. Um, the next full 12-month year, 2024, we increased our sorting services by 9%. And then, if you'll notice up here, we the Tyto absolutely surpassed our droplet sorting services by it's now it was 50. I was shocked when I did this analysis last week for this presentation. I was like, wow, 57%. I hadn't even realized it was that much. So I mean, again, I don't know how much more compelling information you need. I have more testimonials from my customers to share if you do, but that's a pretty darn good story to tell. Obviously, this year's only 5 months into it, so we've kind of leveled off so far. But there's a pretty good competition there, and it's been impacted by our latest in September. Last September, we added the Fax Discover S8. This is not the time to talk about that, but it definitely has an impact on the percentage. Whereas those early adopters of the Tyto who are curious and innovative, they've probably slid over here a moment to see what's this Facts Discover all about. But I have a feeling they'll be back to the Tyto. And again, just as a summary statement of the slide, over the 3 years we've had the instrument, we have accumulated 62 total users out of an average of 200 users of the entire services of the core. And of those 62, we have trained 39 of them to be 24/7 independent. I don't know how many cores can boast that as far as their droplet sorters. I know we only have about less than a dozen. And that's over a long period of time with attrition, taking some of them out because we're an academic institution and people come and go all the time. Okay, so I alluded to a little bit ago that because of all that compelling information, we got our users saying, we want more, we want more. I was presented with an opportunity last May to 2025 to do a beta test of the new generation MACSQuant Tyto Lux. Fortunately, through the initial experiences of the original version, I was able to get opportunities to talk to the engineers and the product managers at Miltenyi. And let me tell you something, they listened. Everything that I asked for, that I said, hey, I love this system, but it needs X, Y, and Z. They went to work and they really knocked it out of the park, in my opinion, because in addition to all those awesome features that the original one has, they've added in an additional 552 laser. And so, yay, my M Cherry expressing cells are able to be sorted efficiently. I'm so excited about that. They've increased the fluorescence detection channels up to 12, increasing flexibility of choices of floors. Complexity of the panels is more highly supported. We've got much more improved signal resolution and higher throughput capability because of the signal processing enhancements. And my favorite, the enhancements in the software that allow us to do standardized workflows. And I would tell you, because Bethany, my crack cell sorting go-to staff member, she provided me with this feedback for this slide with the Max Quantify 3.2 upgrade. The software enhancements include automatic cartridge priming that gives us an a much better experience in processing the cartridges both before and after, because first of all, we can store that magnet, the prep magnet, in a drawer somewhere, which expedites the cartridge prep. And it also lowers the risk of damaging the microchip, that very delicate business end of the cartridge, which then naturally lowers the cost of the expense of this technology. So some of those concerns that, oh, these cartridges are really expensive. Well, they are if you aren't handling them properly. So now we have an argument to say, hey, they're not as expensive as they used to be. Embedded in the Max Quantify software, the new version, are purity masks. So we are able to tailor desired outcomes for the individual's customers a little bit better, depending on what they want. They can get a high yield recovery, edging towards enrichment, or if they need super ultra purity, we can select that mask. It all depends on what the person's downstream assays require. And those masks are now incorporated in the updated Poisson calculator, which makes it even more streamlined of a prep process. And again, my favorite feature enhancement are the automated workflows, including the QC. Uh, um, I'm a big stickler for the performance tracking, and I like it to be standardized, not just for the ease of the staff and the productivity of the staff, but because what that allowed us to also do is simplify the training for new users who are not that savvy on instrument performance tracking. They just need yes, no answers. Is this instrument suitable for me to use? And because we want to be able to create 24/7 access, these workflows allow confidence to be boosted in our users about those performance metrics, even in the absence of the staff being around to bounce questions off of. So let me see here, 24 minutes. I tend to chat. I have several customer testimonies. I decided that they're bonus material. I'm going to use, Gert, if you don't mind, I'm going to take hopefully maybe 2 to 3 more minutes, and I'm just going to really quickly zoom through these to get to the end. Again, these are customers who are all independent users. They have a variety of different target sorting needs. This particular customer, a research scientist, is trying to enrich CAR T transduced cells, whether they're human or mouse CAR T 19 models, for the purpose of in vivo mouse injections. She does an actual mouse model, and then she has a humanized model. And you can see over here highlighted in green her feedback, her study results, and overall impression. Won't read the comments to you. You can see where she's a fan of the technology. What I wanted to point out mostly is after freezing a subset of the human cells, the post-sort GFP positive cells, she thawed them and then injects them later, and she said their functionality is just as good after freezing as they were before. And her previous experience of these same sorts on the ARIA, not so happy. She had a much more difficult throughput situation because of the mouse house hours. She was having a very difficult time on the ARIA doing same day experiments because of the limit on the workflow. But she found with the speed of sorting on the Tyto, she could knock these things out. And she just discovered today that we had the second one. I forgot to tell her. I didn't realize she was doing another sort because she's independent. She comes in when I don't even know she's here. So she's even double happy now. This is, I probably would categorize this independent research scientist. If she could marry the luck, she would. Again, this was a CAR T ex vivo samples from mice. They were injected with BALL cells on day 4 and CAR T cells on day 0. Her intent is to sort the CAR T from the bone marrow and spleen. The big key take home message here is she was able to sort 8 different samples using two instruments simultaneously in less than 2 hours. And then the other key, she submitted those samples for RNA-seq. One of the comments that I wanted to share with you is the not only did the all 8 samples pass the sequencing metrics with excellent assessments, but the bioinformaticians on the project made a point to tell her how easy and clean the data was to analyze. Um, Another feature, accuracy in the sort metrics between the Tyto and the Nexalom cell counter that she has. That's always a bone of contention on the cells of the droplet sorters. We always have differences in those measurements. So she, in her other institution, used to use an ARIA or a Sony droplet sorter for these studies, and she always had less than 80% viability. And with this particular system, these microchip sorters, she's achieving 85 to 95 as a habit. These results from this study down here show you anywhere from 90 to 98%. Can't argue the point. So I think I made my point, and again, I'm way over my time. I can share more of these, but it would cut into Gert's opportunity to share his great experiences. This is another customer. One thing she had she enumerated all the yays in her experience. She did have a comment because she was an old Tyto user. She didn't have experience on the Lux yet. She was only pointing out that the data storage, onboard data storage, and the resolution for mCherry needed improvement. But guess what? The next generation Tyto Lux totally handled all those concerns. So she's good to go for her future studies. So we now have cell sorting platforms for everyone, including the more recent FACSDiscover S8, but more importantly, our second Tyto Lux. And hopefully, even though this instrument with this specific model that's currently on the bench has to get packaged up and moved up to our new lab in July. Hopefully, because I sneaked it in on the bench for the last year, my customers who are staying here at the main campus will get very upset when they see it's been disappeared, and they go to the advisory committee and the powers that be and get us to buy another one. So if we have time later, I can talk about it. I have some bonus slides, but at this point, I want to go ahead and give Gert the floor. And I just want to thank you for the opportunity. I hope I was able to compel you as to the importance of this technology in your core facility or in your own personal lab if you're doing delicate downstream assays.
So I will take over. Jody, what an amazing presentation. Also, I would like to thank Alex and Technology Networks for the opportunity to present today. Again, Jody, very insightful, very inspirational. We are newbies, so we are adapted to technology way later. And yeah, I was really happy to see all the results. So I will take off. So again, very happy to talk today about advancing cell sorting strategies in our core facilities. And I will first start with introducing the institute where I work. It's the VIB, and it's a life science research institute in Belgium with a very entrepreneurial mindset. And we have a vibrant community of highly skilled scientists, and which are very multidisciplinary. So VIB itself, it's one institute, but we are not in one location. So we are spread across the whole part of the Flemish part of Belgium, and we collaborate with all the Flemish universities, so Antwerp, Brussels, Ghent, Leuven, and Hasselt. So to support, so the research at VIB is thematically organized in different research centers. So for example, in Leuven, they focus on cancer biology, microbiology, and brain and disease research. But my core is located in Ghent, and there we have three large VIB centers: the VIB Center for Information Research, the Center for Plant System Biology, and the Center for Medical Biotechnology. And to support all this ongoing research at VIB, VIB has set up a technologies program, and this is a website where you can go to learn more about the different core facilities that we have. So we clustered them a bit into cellular biology assays being flow cytometry, single cell technologies, bioimaging. Next to that, we have a technology scouting team. There is some focus on research data services. We have the traditional omics technologies, etc. And I made a short screen recording because it's a very nice website to go to. So here we have our protein production and nanobody production core facilities, the omics technologies, and then of course my favorite technology, cytometry. And also here you can visit our core, but also our partners in the University of Leuven. So feel free to have a look at it and to learn more about what we have on offer. So this brings me to my course. So the VIB Flow Core in Ghent, we have a team of 6 people. So from the left here on the picture is Katrien and Julie, both flow cytometry experts in my team. In the middle, we have Eileen, who is a lab manager of the team, and Margot, who is a shared lab technician between the flow core and the single cell core. Me heading the team, and recently also Sophie Van Gassen, last year, December, joined our team, and she is well known in the field because, for example, of the development of the FlowSolve algorithm and also the QC algorithm. So, what is our mission? As I think the mission we have is, like many core facilities worldwide, is to provide quality service and give our users access to state-of-the-art equipment. So, how do we do that? We have specialized staff fully dedicated to flow cytometry, and we make sure that our machines, our flow cytometers, sorters, and other devices that we have in our core are, of course, in perfect shape and quality controlled. We put a lot of effort into training, not only this generation, but also the next generation of scientists. We get many students visiting our core every year. And we offer a full theoretical training program and, of course, train all our users on the devices itself. So we help them with designing, executing, and analyzing their experiments, or we help them at particular parts of that experimental pipeline. At VIB, there's always a strong focus on new assay development. So we help the research teams developing these new applications. And when we see something novel, of course, or new technology popping up, if it's of interest to our user base, we will implement this technology and bring it into service mode. So our core in numbers, we are quite a large academic core. So we have currently 13 flow cytometer analyzers and 8 FACS cell sorters. We have an average user base of around 280 active users and booked usage time of around 9,000 hours every year. and perform more than 1,000 cell sorting experiments per year. Again, as mentioned, we do some training, giving a lot of introduction to flow cytometries, but also, for example, we have our computational cytometry summer school, which is just finished, and it was already in its third edition, so it's very international training that we offer here in Ghent. So, but I'm here today to talk about what we have been doing with the MACSQuant Tyto cell sorter from Miltenyi. And we were involved in the beta testing. So we were not owners of a Tyto yet. And we were approached by the Miltenyi team beginning of the last year. And we were intrigued, and by listening to all of the new things that were onboarded now. So we were doing the beta testing between February and May last year of this new MACSQuant Tyto Lux sorter. Many things already mentioned by Jody, but I will do it again. So what is really interesting to us was the addition of this fourth laser. So next to the red, blue, violet, we now also have yellow laser on board, which is of course very interesting for red fluorescent protein excitation. Next to that, also Miltenyi has a new type of cartridge, which was of interest to us and to our user community, which are the large cell cartridges. I will come back to that later. And then in the software, there is now the option to go for different sorting modes. So you have the option to go more for yield sorting, or if you want to have more pure sorts, you can do that. Next to that, some improvements like automation in terms of the door, automated door and loading chamber, automated priming of the cartridge, and some extra software improvements. So overall, the beta testing went very well. So we had a very, very successful demo for a couple of months, and technically, we didn't got anything else than positive feedback from all the testers. So there were multiple VIB centers. So the three large centers in Ghent were all involved. So we had a large variety of and diversity of samples, not only in terms of species, but also in terms of cell types. We had a really fantastic collaboration with the Miltenyi team. Really a big shout out to them in terms of application support, talking to our users, discussing what we could do better or how we should change stuff. Very good technical support, and also these online feedback sessions were really great. So after the beta testing period, which was really successful, we went into the summer, and we really decided to start up the acquisition process, and we managed to secure budgets, and we acquired the system in September 25. So here, just sharing on the right side some of the LinkedIn posts. So we also officially launched the system with so it was really nice. So what did we test? So I will talk a little bit more about them. So we tested different samples, eosinophils, hepatocytes, neutrophils, macrophages, mainly from murine samples. Also nuclear sorting, both on plants and murine, and of course quite a lot of different cell lines, being human cell lines, mouse cell lines, to focus on this red fluorescent protein excitation. So here there is just a short, it's picture of how the cartridge looks, and for people that have never seen it. So here on the left side, you see the input chamber where you will put your pre-sorted, the sample that you are going to sort will go in there. There is a kind of agitator and also a 3D filter to make sure that the system will not clog. So all the cells go into the bottom, and there is the microfluidic compartment where your cells get aligned, struck by laser light, and then there is a very short magnetic valve which will take out the cells of interest. So your target cells will end up in the middle of the positive sort chamber, as they call it, and the cells that were not of interest to you will end up in the negative sort chamber and can be sorted again if you want to take out another population of interest. So I made a video. It's a little bit noisy, so apologies for that, but I think it's really nice to show you. So I will talk a little bit louder. So yourself enter here and so you can click on a laser and it's technical. And here there is a magnetic valve. You can see something up here and all the sides to be down, and that's what and that difference for there. Can really think that we're trying to find the are kind of fun to look at. Um, so why were we interested in this new Tytolux device, cartridge sorter? For a couple of reasons. First of all, again mentioned by Jody, is this microchip sorting. which is of interest to many of our users. It's patented. It can do gentle, sterile, aerosol-free sorting. So it's a lot of boxes to check, and they really preserve the cell integrity. So again, it now has 4 lasers, the Tytolux. We have up to 12 fluorescence channels, which allows for a more flexible panel design. Also, the software is doing that quite nice. It sorts at really high speeds and really good, very high purities. So maximum flow rates can go up to 110,000, although we did not try that. But the magnetic valve can really actuate at 30,000 actuations per second. So and reaching very high purities. It's quite user-friendly to use, and the whole system is temperature controlled, so you can work at the temperature that you want to work with. And what also is for me very tempting is the low input workflow. So it means that you can really work with low input volumes, but also everything that gets sorted. So the sort fraction ending up in your positive sort chamber is super, I would say, concentrated, and it does not a lot of volume. So it's very interesting for downstream assays. So you don't need to spin your cells down anymore. So especially interesting for 10x genomics or BD Rhapsody, this is for me a real game changer. So for us, it really complements what we have. So it complements our existing infrastructure of high-end droplet sources that we have available to our users. And we made this decision to purchase after the successful testing. And there was really a buy-in from all the 3 centers, which was really unique. And also we topped it up with some VIB technology fund. So today I will share some of the user examples, and let's start with the testing of the yellow laser. So there are 3 laser modes. So you have blue, red, violet, which are always on, but the yellow you can play around with it. So you can turn the yellow off if you're not interested in using it. You can turn it on, or you can use a kind of balanced laser modes. And the reason why you have these modes is you have to realize that the yellow and the blue laser share the same pinhole, so they are co-aligned. So this is important to take into account. What we noticed is that the yellow laser, when turned on, it excites the red fluorescent proteins much better. And we did a variety of cell lines, also a variety of proteins, cherry, TD tomato, RFB. So we noticed always better excitation and better emission, of course, better resolution. What is interesting to take into account, or what you should take into account, is that the side scatter drops when you work in the mode where the yellow is on. The reason because of that is your yellow laser is shining at 100 or working at 100%. The blue is tuned down to 10, and the blue is linked to the side scatter. So just take that into account. And this is why we also more prefer the balanced option, where the yellow and the blue laser go into some kind of 50-60% kind of intensity, and this works really well. So on the right side, 2 examples I will give. So this is the B16 mouse cell line, which is expressing mCherry. So if you don't have the yellow laser on, there is some excitation, but not a real good. So it's coming from the blue in this case. So your mCherry gets excited by blue laser, and you will see a little bit. But if you turn the laser on, of course, resolution becomes way better, and it's really like a separate clouds. Side scatter went down a bit. So in balanced mode, this is fixed. You don't see the side scatter effect that hard, and you really have a nice population. So here the same for the TD tomato cell line is the same example. Yellow off, really not a good excitation. Turn it on, you really get the separate clouds, so easy to gauge. And in balanced mode, you're still in a very good mode to work. So next to that, we tested the new option of the purity modes, which is, of course, I think very, very interesting to have on board of a microfluidics device. So there is the option to go from ultra yields, high yields, to high purity and ultra purity. And on top of that, there is an additional checkbox to make sure you take out the coincidence aborts, so that coincidence events get aborted. So what we did is we tested these different sort modes with, again, mCherry positive cell line, and we checked always the purity on a flow cytometer analyzer. So the first test we did, they were all super high. So it was in all modes high, like more than 98%. And just as a remark again, so the coincident board checkbox is really nice, but be really careful with it that you don't lose too many cells because of the stringency that you put in there. So this is the cell line we're using for side scatter, some back scattering. And here you see that we have a cell line which is 72% MCherry positive. And these are the results from the first test. So going from ultra yield to the most stringent ultra purity with coincidence abort. And we go from, I think it's hard to read here, from 98 to 99.9. This all looks good. Maybe it's too good. So we realized we should increase our input a bit more. So that's what we did. So the input sample, we increased the concentration and we did another experiment. So with 10 concentration of 10 million cells per ml. And the input was not in the best condition, so we'll be honest about that. So it was a mix of the parental cell line and the B16 TD tomato cell line. They were already 3 days in our fridge, but anyway, we gave it a go. So we respended the cells in Tyto running buffer, and then we checked first the cell line on the analyzer again. We noticed that around 20% were TD tomato positive, and we loaded these cells on the cartridge, and we got really a high trigger rate, about 20,000 events per second. And now you see that the purity mode starts doing a good job. So we never got like super high purities, but it all makes a lot of sense because you really put in a lot and you make the machine work very hard. And it also warns you that technically you're going too fast. So the software gives you a warning that you should be dilute the sample a bit more if you want to get the highest purity. But again, very impressed by the numbers we saw there. In terms of gentleness, we also did some experiments together with, in this case, Sjoerd Schetters, a postdoc from the Bart Lambrecht team. Here in our institute is a group studying asthma, and they struggle a lot with eosinophil sorting. So they eosinophil sorting on a traditional droplet sorter. So you can sort them quite pure, but after sorting, you'll also always see that. So here are your eosinophils. That a lot of them basically didn't survive the process. So also on this website, on the Technology Networks platform, you can look for this webinar, which is recorded, and it's short explaining what he did. So I will just go quickly through it. So he sorted from asthmatic mice after he did a lavage, a bronchoalveolar lavage, which when you have around 60% of eosinophils, he enriched them to up to 99% pure eosinophils, brought them in culture, and the survival or the post-culture viability was really, really great. And he had let's say he was very happy user. And this was some feedback we got from him. So he liked the gentle sorting. He liked the fact that you could really put in a high volume. You don't need to spin. And also the fact that the low volume output was really tempting for downstream proteomics or transcriptomics assays. Super happy with the purity and also with the speed. And of course, also with the ex vivo culture capabilities, like in terms of sterility, viability, and also functional things that can still be performed afterwards. So also here he mentions the ease of use. A pilot he did also was sorting eosinophils that were expressing TD tomato. And then what he did is he sorted them from this transgenic mouse to very high purities, and then he did transfer of these eosinophils, 500,000 intratracheal in another mice, which was also expressing yellow fluorescent proteins, eosinophils. And after 1 day, when he sacrificed the mice, he could retrieve all these, basically both populations. So it was the first time he was capable of doing that. Another experiment from the same team, not from Sjoerd, but from the same team, because they were very triggered by what he was doing. So they sorted IV labeled versus non-intravenously labeled eosinophils. A little bit more difficult in terms of gating strategy. But the purpose here was really to make sure to do single cell RNA sequencing on 10x genomics. So we know the sort was very successful. We did all the purity checks, but we don't have the sequencing results yet. But the goal is here basically to distinguish the circulating eosinophils from the tissue resident eosinophils in the lung, and to learn more about that. Another take on from another team, a little bit different, is where they use the MACSPlex EV kit Neuro, which is a new kit from Miltenyi, and they use the kit a little bit different than it's supposed to be used, but then it's really cool EV sorting. So they used the kits. I for the sake of time, I will not go into depth a lot, but it's technically a cytometric bead array kit where you have these capture antibody beads labeled with different intensities of PE and FITs, and they have 37 different antibodies attached to them. You load them with EVs of interest, your EVs of interest, and then the EVs will bind to the antibody capture beads. And then you can do a readout if you want with a cocktail of detection antibodies, in this case, CD9, 81, and CD63, which are like universal EV markers. But what this team did is they took out a really specific subpopulation and they sorted it, which I think is really cool idea. And they are now looking into that more in depth. Next to that, as mentioned, we also have a very large plants biology, plant systems biology research center. So we have many researchers doing plant flow cytometry. So we did quite some tests with Arabidopsis thaliana, the model plants in the plant research field, but also maize, soybean. So we did a lot of protoplast sorting, also nuclear sorting, and we, in this case, really tested the large cell cartridges versus the high-speed cartridges. Also, other tests are currently ongoing with ATAC-seq, very successful. First time we can do this successfully. And what they do in many cases is they have the plants, they will chop the plants and also add some enzymes to get rid of the cell wall to end up with single cell protoplasts. And then they will use sorters and combined with BD Rhapsody or 10x Genomics to generate these plant single cell atlases. So here, just some examples of what we are doing here on the left again. So TD tomato positive protoplasts. On the left side is Colombia, is the wild type. And here you see the TD tomato signal. We sorted these protoplasts and analyzed them afterwards on confocal and also for downstream RNA transcriptomics. Also here, large cell cartridge. It's not super clear, I know, sorry for that, but we have seen really amazing results where we see that we can now enrich, also we can enrich for larger protoplast, which is not possible with the systems we have available here on our traditional sorters. Other ongoing experiments are the double emulsion droplets. This is more exotic. It's oil water droplets. And again, same results. They are loaded with Alexa 700, so pre-sort and post-sort, again, analyzed afterwards on flow cytometer analyzer, and we can really see great enrichments. Same for the bacteria. Here we have E. coli expressing RFP, and again, very high purities afterwards, very fast sorting. And here, as the last example, I have mouse liver nuclei. So where we started with a pre-sorted fraction, and then this is the post-sorted fraction in high purity. It's very high. Don't The reason why we get this background and it's only going to 70 is because we sorted in this buffer. So the buffer contains some BSA, so you will always get some, I would say, noise from the buffer in the background, but pretty sure that this population is around 95% pure. And with that, I just will stop and acknowledge, of course, my team. Also, Janto Malvliet, who is a student doing a lot of the tests on the Tyto, and all the researchers involved, and not to forget, because it's not on this slide, but the team from Miltenyi for their amazing support. So especially Adam Serva and Sudhir and Alina and Frank, you know who I'm talking about. They really helped us out a lot. Thank you for your taking some time to listen to this story.